Here we go with a second step of the central dogma. So in the last video we looked at DNA replication, so how we can take DNA and make copies of it if we need to replicate the cell. And now we're going to look at the step from, let's go from DNA to RNA. So this is called transcription, because really what's happening is it's being transcribed, it's being copied. It's still in the same language, nucleic acid, a couple little differences, but we're getting ready to export it out of the nucleus into the cytoplasm so that it could be made into protein in the last step. So transcription is the conversion of DNA to RNA. And if we look at the little diagram right here real quick, there's a couple areas that we're going to focus on. The first, and maybe the most important, is there's going to be a section of the DNA that's the coding region. This is going to code for the RNA that we actually want. Now, just prior to that, there is going to be a promoter region. This is kind of the start signal. This is a sign on the DNA that allows RNA polymerase and several transcription factors to bind and start the whole process. And then finally, at the end of it, we've got a terminator region. This is going to tell the RNA polymerase stop, unhitch, and this will be the end of the production of an RNA transcript. And specifically, at the end of this process, what we're going to get is something called pre-mRNA, where the M stands for messenger. Now, here's a little bit more detail of that. So if we look specifically at the promoter region up here, and in that promoter region, there's a sequence of DNA, and in there is something called the TATA box. So it is a sequence of TATA, and you can see that again, it's anti-parallel. So we have TATA in one direction on the top strand, and then we have it in the opposite direction right underneath that. That's going to serve as the signal for these transcription factors to bind. And once the transcription factors bind, other proteins come in, and finally the important enzyme, RNA polymerase, comes in and forms what's called a transcription initiation complex. So it is this complex of polymerase and other proteins that's going to allow the DNA to be unzipped, mRNA to be copied, and then we're going to close the DNA back up. So when we looked at replication, we talked about how there's helicase and ligase and polymerase and primase, all these different enzymes. And in transcription, it's really all-inclusive. They're all included in one complex that will go through and carry out all the processes at once. So here it is blown up. So here is our transcription initiation complex, the large bubble around the outside, and the major enzyme of that being RNA polymerase. And so what it does is it starts at a section of the DNA, it unwinds it on one end, over here on the right, and then it brings in complementary RNA nucleotides and adds them in. Now remember, what's the main difference between DNA and RNA? There's one base pair that's different. So in DNA, wherever we would have a thymine come in RNA, we have a uracil instead. So if we look right here, matching up with the adenine on the DNA will be a uracil from the RNA. And again, it's being copied from the 5' to 3' end, just like we've seen with all nucleotides. And we get a single strand of RNA, and then over on the left, the DNA is wound back up, and we get our primary transcript. We get our new strand of RNA from that. And so it basically kind of breaks down into 3 steps. The first step being initiation. So that's when we get our transcription initiation complex. I'm going to write TIC. That will bind at the promoter region. We've got our elongation phase, where the transcription initiation complex is moving along and making our primary transcript, our RNA. And I know that light blue color makes it hard to see, but it is there. And finally, we've got our termination. That's where the transcription initiation hits the stop sequence. It disengages, and we have our RNA that's ready to be processed. So it's relatively straightforward compared to DNA replication. There's fewer steps, and we end up with a single strand of RNA. Now we call that our primary transcript. And here's kind of a summary of the whole thing, where starting over on the right-hand side, we see the unwinding of the DNA. In this picture, the pink is the RNA polymerase. It adds the complementary base pairs in here in the middle. And again, remember, it replaces thymine with uracil. And we get our mRNA out, and it is wound back up at the end. So this is a good diagram to kind of summarize what happens as we go through. Now let's look what happens to that strand of RNA. Several things have to happen before we can export it out of the nucleus. So if we look at the box in the upper left-hand corner here, here is what's referred to as post-transcriptional modifications. So transcription has already taken place. We've got the RNA, but we've got to modify it some. We've got to change it a little bit. And the first two things that happen is we put caps on the end. So we put a 5' cap, and we put what's called a poly A tail on the 3'. And the main reasons for doing that, number one, is protection, so that the ends of the DNA, or excuse me, the RNA don't get degraded, but also there's some signal recognition. As it leaves the nucleus, it's going to provide kind of a passport, if you will, where the nuclear pores will recognize it and allow it to leave the nucleus. So we've got to put a 5' cap and a poly A tail on that. And then the other thing is we have these regions of the DNA called introns. And they're kind of non-coding DNA. We don't really want to pass them along when the RNA leaves. So what we have to do is we've got to splice them out of there and remove the introns, and then we splice the exons together so that we get our final mRNA that will leave the nucleus. And I'll come back to this diagram in just a minute, but what I want to show you what happens, how do we get those introns out? And I love this just because of the name. So what happens is there's a group of proteins that get together called a spliceosome. And so what they do is they recognize the ends of the introns, so right here and here, and they grab hold of it, they bring it together, and they incise, or they cut the intron out, they pull the exons together, and we get a spliced version of the exons. So you can see in this case there's only 2 exons that are cut together and put for that. So let me go back to the previous picture real quick. So that's what happens right here when we remove these introns. So that's part of the post-transcriptional modification. So now we're at a point where we've got mRNA that can be exported out of the cell. It's ready to go to the cytoplasm and carry out the last step of translation. And that's where I'll end this one. And again, just like last time, I've got a quick video or animation to show you that'll make it a little bit easier to follow, because I know sometimes my pictures and my explanations aren't the best. So let's take a look at this one just like last time, and hopefully this will clear things up a little bit.
The synthesis of messenger RNA is called transcription. Transcription begins when RNA polymerase recognizes and binds to the promoter region on the double-stranded DNA molecule. A particular subunit of the messenger RNA, called the sigma factor, participates in recognition of the promoter region. Soon after transcription is initiated, the sigma factor dissociates from the RNA polymerase. RNA polymerase moves along the template strand of the DNA, synthesizing the complementary single-stranded messenger RNA molecule. Synthesis is in the 5' to 3' direction, with new nucleotides being added to the 3' end of the growing messenger RNA molecule. As the RNA polymerase advances along the DNA, it melts a new stretch of DNA and allows the previous stretch to close. When RNA polymerase reaches a specific sequence of nucleotides on the DNA, called the transcription terminator, a hairpin loop structure forms in the messenger RNA, causing the RNA polymerase and the messenger RNA to dissociate from the DNA.
And there we go, and hopefully that clears up a little bit of the process. I just want to go back and show you guys one thing that I neglected to talk about. So on here in the picture, it's right here in the picture, they'll talk about the template strand or the sense strand. So that's the one that's actually being copied, because when the DNA gets unwound, there's two different strands that could be copied. The one that gets copied is called the template strand or the sense strand, and the other one is uncopied, and different genes will copy different sizes for that. So there we go. That will wrap up our transcription, and we've only got one step to talk about when we come back, and that will be translation.